Triple negative is one of those breast cancers that keeps oncologists up at night. It's aggressive, it's hard to target, and it comes back with a vengeance. Even after successful surgery and chemo, patients relapse on average after 3 years. Now, every cancer is different, so why shouldn't every cancer treatment be different? This is what BioNTech, yes, the same COVID vaccine people, tackled in their most recent phase 1 clinical trial that they just published. They gave 14 triple-negative breast cancer patients a personalized cancer vaccine, and 11 of those 14 patients saw no relapse after 6 years. And that's genuinely impressive. But those other 3 patients, those are the ones we gotta talk about. What's up, nerds? Now, before we get into the details, I have a question for you. Do you like my new lab coat? My friends made it for me. Gotcha! Quick and important note on triple-negative breast cancer and why that was chosen as the patient population to make this vaccine for. You see, most breast cancers have these cells that have these little molecular handles, surface-bound proteins that make it easy for most treatments to bind to them and eventually kill those cancerous cells. These general class of treatments are called targeted therapies. Now, there are 3 specific handles that most breast cancers have at least one of: the estrogen receptor, the progesterone receptor, or human epidermal growth factor receptor 2, also known as HER2. And there are a number of targeted therapies that are available for any of these targets. They're specific, effective, well-tolerated, and very well studied. But triple-negative breast cancer has none of these handles. No handles means no targeted therapy. And this is a real problem during metastasis, or when the breast cancer cells migrate to other parts of the body. It is incredibly difficult for treatments to find and kill these cancer cells effectively. The standard of care for such cancers is still chemotherapy. This blunt force, non-specific treatment, like dropping a nuke on the whole body just to kill 1 or 2 baddies. And it's only marginally helpful. TNBC on average still comes back after 3 years of successful treatment. And this is the gap that cancer vaccines fill. Like a highly specialized cleanup crew, you train your immune system to detect any cancer cells that are still floating around after the main tumor is gone, reduce the risk of a new, deadlier tumor forming. But making a vaccine against cancer isn't that easy, because we have to understand what makes cancers so personal. You know those little handles we talked about earlier? Every person's cancer will display a different array of them to your immune system. Not just 1 or 2, but up to 20 different surface proteins that, in theory, your immune system can detect. Proteins that aren't showing on your healthy cells. And they're all encoded by the cancer cell's DNA. Okay, but there are hundreds of cancer-specific genes. How can you know which ones this single person's cancer has? For that, we have to talk about next-generation sequencing, or NGS, one of the most advanced and quickest ways to assess a cancer cell's entire genome. Basically, researchers take both healthy and tumor cells from a patient, isolate their DNA, and then chop them up into little bits that can easily be identified. These bits are amplified and run through a number of programs that can analyze the quantity of each DNA strand. Finally, they complete the genetic jigsaws of both the healthy and tumor cells and compare to see which sequences are in the tumor cell that are not in the healthy cells. Think of it like running 2 simultaneous spell checkers. The DNA words, if you will, are mapped side by side, and the spell checker goes in and circles all of the mutations that are only found in the tumor. There's no need for those big 3 handles. These are your new targets, and they are called neoantigens. So in this paper, they did NGS on all 14 of their triple-negative breast cancer patients. And they found most patients had anywhere from 15 to 20 different neoantigens, which is both terrifying and exciting. As much as that's a lot of cancerous mumbo jumbo, it's also 15 to 20 new theoretical targets per patient that this cancer vaccine can be based off of. And just like with vaccines for infectious diseases, the point of the cancer vaccine is to show little bits of these neoantigens to a patient's immune system so that they can learn to beat the absolute wheels off of this cancer. So listen up, molecular biology nerds, this is where the engineering gets really cool, because we're gonna get into the nuance of how this vaccine was manufactured. You're welcome in advance. It is incredibly important to express as many neoantigens in a single vaccine dose to give your immune system a robust enough data set to train on. So they encoded each patient's neoantigens into a single engineered mRNA strand. Yes, that's right, 20 things on one thing. That's tailor-made to their cancer-specific mutation profile. It being an mRNA vaccine is no accident. It is crucial for this whole system working because it's the fastest way to make a vaccine. In this trial alone, the average time to go from biopsy, so learning anything about that person's cancer, to a finalized mRNA vaccine was just 69 days. Nice. We're talking several months faster than other traditional vaccine technologies. Speed is of the utmost importance when you have to make a new vaccine for each patient. But the number one benefit of using mRNA is that it's programmable. Just change the sequence based on that patient's tumor, run a very similar manufacturing protocol, and you get a different personalized cancer vaccine, either for a new patient or for a new tumor that's developed in the same patient. More on that in a little bit. The manufacturing fun doesn't stop there because you can't just administer it for an mRNA into your body. It'll chop it up pretty much immediately. The delivery vehicle they use is called an RNA lipoplex, or LPX. It's essentially a bundle of finely curated fats that package the mRNA and safely carries it through the bloodstream. And you might be thinking, this must just be leftover lipid nanoparticle propaganda from the COVID days. Hey, actually, can you come here for a second? We're gonna go touch some grass. There are several key reasons why this formulation is good, specifically for cancer vaccines. First is the simplicity. Just mix the RNA with certain types of lipids and let the electrostatic interactions guide a nanoparticle formation. I'm oversimplifying it a bit, but it genuinely is much easier than any of the other vaccine technologies that we just described. It complements the speed that comes with making mRNA. And the lipoplex is meticulously designed. It's optimized with specific lipid ratios to get picked up by dendritic cells all throughout the body. These dendritic cells decode the mRNA that's inside it, manufacture the neoantigen proteins, and put them on display for your T cells to read. The T cells clock those neoantigens as the enemy, and they start hunting. The second is the route of administration. You see, most COVID vaccines were just administered intramuscularly. They initiate an immune response just in your arm, but this one is delivered intravenously. It's trying to deliver that neoantigen code all throughout your body. This way, it increases the chances of your immune cells to find that neoantigen code close to any cancer cell circulating in the body. Simple, yet elegant. But is it effective? And here's where we finally get to talk about the clinical data. Their phase 1 trial looked at 14 triple-negative breast cancer patients, all enrolled within a year of finishing standard chemo and surgery. Each patient was given 8 doses containing 50 micrograms of their personalized mRNA vaccine. Because these were used as a cleanup crew, these authors were mostly focused on rates of relapse and the different immune cell types in these patients to determine different mechanisms of action. And what they saw was nothing short of incredible. 11 of those 14 patients had no relapses after 6 years. Remember, that's almost double the average of other patients with TNBC. And the effects weren't short-lived. Even though the vaccination stopped after day 64, they found 2 different T cell populations that were still ready to fight the cancer at any given moment years later. These are the kill now T cells as well as those memory T cells. They're like the elephants, they never forget. And here's what really tipped my hat to these authors. They didn't just rest on their laurels and focus on the good, these 11 patients. They actually spent the majority of this paper focusing on the bad. Those 3 patients who did relapse, trying to figure out how they could make their personalized cancer vaccine better. Each patient tells a different story of why this cancer vaccine didn't work. And so in these authors' honor, we're gonna go through each one. Patient number 1 is the most interesting to me because at first it seemed like they just had the weakest immune response out of all the other patients. Or so they thought. To try and stimulate this person's immune system some more, they gave her something called a PD-1 checkpoint inhibitor. Here's how that works. PD-1 is naturally found on your T cells, and it acts as a brake. It tells it to slow down and not attack certain cells in the area. Cancer cells are really good at manipulating this and usually tell it to turn off anyways. So by inhibiting PD-1 or inhibiting that inhibitor, they were able to reactivate the immune system. This led to this patient eventually going into remission. They gave clear signals that some people need combination therapy. They need an extra little step to get the response they're looking for. The vaccine left some breadcrumbs. They just needed the right environment to snatch them all up. Now, patient 2's cancer was trying to be a little sneaky. They found that she had low expression of something called MHC class 1. If the neoantigens are the flags that signal to your T cells there's a problem, MHC class 1 is the flagpole. This patient's cancer cells had no flagpole, basically trying to hide itself from the circulating T cells. The real life implication of this is sometimes the T cells are primed and ready to go, but there needs to be real-time monitoring of MHC class 1 to make sure they're getting the appropriate signals. Now, patient 3 is probably the most terrifying one for me. The cancer vaccine actually worked at first. She eventually relapsed, and when they biopsied that tumor, new antigens formed from brand new genetic mutations. The reason why this is so scary is it's a classic example of how cancers are so adaptable. Even a personalized treatment regimen isn't enough to stop cancer from inventing new ways to try and kill you. Now, there are ways to improve on this, maybe having broader neoantigen coverage right from the get-go, or developing newer, quicker ways to actively monitor what neoantigens need to be given, but it's something we'll probably have to wait a few more years until it hits the mainstream. Regardless, I'm still very happy that this paper came out. It gave me a whole lot of new perspectives on the nuance of personalized cancer vaccines, not just from the chemistry and the engineering side, but from the clinical side as well. Mind you, this is just a phase 1 study. It really is just looking at a baseline safety profile for these treatments. They'll still need a few years to do the phase 2 and phase 3 studies to determine efficacy before they're approved for public use. Overall, I'd still give this paper a nice 8.5 out of 10 on the old nerdometer. But based on what you've seen, what would you rate it? I think I know what the American Food and Drug Association would rate it. It's probably like a 1, because for some weird reason, they're still demonizing mRNA vaccines for pretty much no scientific reason. If you want to learn a little bit more about the current trials and tribulations that similar companies like Moderna are going through on that, you can click on this video here. But that's all for me today. Thank you all for stopping by, and you stay classy, nerds.